Hello, my name is Nicholas Seinoff. This semester I worked with Austin, and our research focused on the study of HeLa cells. I'm going to keep you at a zoomed out look right now while I give you a brief introduction. HeLa cells were the first ever immortalized cancer cell line. This was momentously important in all sorts of scientific research. One benefit to immortalized cells is that scientists doing research don't need to keep getting more living cells from a living source every time their cells that they're using inevitably die. With HeLa cells, they keep dividing and dividing indefinitely, so cellular research is much easier and cheaper, and this basically happened overnight. Now, this led to the discovery of a polio vaccine. It led to better understanding of salmonella. It led to better understanding of the effects of radiation and X-rays on human cells. And the accomplishments of HeLa cells are endless. And so it's exciting that we got to work with HeLa cells in our research here. And so I'll get a brief I'll give you guys a brief introduction of the biology. Right here is a phase contrast microscopy image of the HeLa cell morphology. Notice that they're all kind of bunched together. Um, so HeLa cells, they are an anaplastic cell line, and they lack the estrogen receptor, the progesterone receptor, and the human epidermal growth factor receptor. And in this semester-long research, we tested the cell line for its proliferation rate via a cyclone assay, its mitotic index, its anchorage-independent growth, and its reaction to chemotherapy treatments. So let's scroll up here to the first of our studies, the cyclone assay. And I'll zoom in a little bit. So, for our first experiment, we set up 4 different samples via trypsinization and cell counting via a hemocytometer in order to get 1,000 cells in each well. We let them proliferate for different time periods, 4 hours, 1 day, 2 days, and 4 days. And we let them incubate for that long. And then once they sat for that long, we froze each one, thus killing the cells and stopping them in their tracks. And letting us get a good look at them. This allowed us to measure the amount of DNA or number of cells in the sample using a fluorescent microplate reader, which is basically a spectrophotometer. We were able to get values consistent with the amount of fluorescence emitted from each time point sample. We normalized these samples, giving us a fold change with regard to proliferation. So as you can see on our single line graph, that is HeLa's proliferation rate of increase. And as you can see, after 3 days, the HeLa cells multiplied to 5 times the amount of the cells in the original sample. Now, compared to the class's data right here, without SW620, because it skewed the data because its proliferation rate was so high, the HeLa cell line ranked 3rd among everyone's proliferation rate within 4 days. So we can conclude that HeLa is relatively proliferative. Next, we move on to the mitotic index lab. And so you see here, we found HeLa's mitotic index through fluorescent microscopy. We seeded our cells onto cover slips. We killed and stained the cells, and we stained them for actin and DNA. And so using fluorescent microscopy, we were able to see which cells among each of our snapshots were in mitosis. So if you look at this picture right here, I know it's a little small, but the red fluorescence represents the actin, and the blue fluorescence represents the nuclei. And so basically we were looking for condensed nuclei in any process of mitosis. And so these nuclei were illuminated further. And we did this with 5 different pictures. We counted up the amount of cells in mitosis, compared that to the amount of cells total in the picture, divided those numbers to get a mitotic index of 4.9% for HeLa cells. And as you can see in this graph, we compared each percentage of the class. HeLa was the fourth highest among all the cell lines. From my research, cell mitotic indices can vary from 2 to 20%. It varies from cancer to cancer. But from journal articles on cervical cancer, I found that any mitotic index higher than 3.5% needs chemotherapeutic treatments. So, hypothetically, if a patient had a mitotic index of 4.9%, chemotherapeutic treatments should be recommended. Next, we move on to the Anchorage Independent Growth Lab. This is the first lab we had to do online due to COVID-19, and so we had to use MDA 435 as our cell line. They are similarly aggressive, and they are a breast cancer cell line, and they mimic HeLa's receptor lacking of receptors. To measure anchorage independence, we put cells in U-shaped wells. This way they couldn't anchor to the bottom of the well. And we started with 1,000 cells in each U-shaped well, and we let the spheroid proliferate for 14 days. We collected images and measured the tumor spheroid using imaging software along each set of days. And then, as you can see by our graph in the pictures, the tumor spheroid grew consistently and significantly over the 14-day period, proving that MDA 435 can proliferate without anchorage dependence. Okay, now moving on to the last lab, our chemotherapy lab. So in this lab, we tested the effect of different chemotherapeutics on MDA 435. We tested 6 different treatments: cyclophosphamide, cisplatin, paclitaxel, methotrexate, tamoxifen, and a combination of 3 of the drugs listed here. We did this via a Cyquan assay, which we've already done. We tested the proliferation of each treatment with the MDA 435 cell line, each starting with 1,000 cells respectively. And so these are our results. And so the most interesting results from the study seems to be that I suspected that the 2 alkylating agents, cyclophosphamide and cisplatin, would be similar in their treatment, but CP, as I'll abbreviate it, had a negative effect on the cells, where cisplatin worked well and didn't let the cells proliferate. And so I did some research, and I found a published article that said that cyclophosphamide can actually promote breast cell proliferation. However, it's not entirely understood, so I'm not entirely sure why this outcome came about. And initially, I suspected that tamoxifen, which I have highlighted here, would work well, but it also proliferated 9-fold, which is not great. And so why would a drug that is specifically designed for breast cancer cell lines not work for MDA 435? Well, MDA 435 lacks an estrogen receptor, and this cancer treatment is designed to compete with estrogen in binding to estrogen receptors in order to slow proliferation. So because this cell line doesn't have estrogen receptors, the drug didn't function properly. Another thing worth noting is that the combo medication performed very well in limiting proliferation. But as an oncologist or a doctor, I doubt that they would recommend a combo treatment if 2 other drugs, cisplatin and paclitaxel, work just as well. So that you probably wouldn't recommend a combo drug because the side effects can be significant. In conclusion, it's been a bit of a weird semester. We had to switch from HeLa cells to MDA 435 cells in our experiment, but ultimately the cell lines are similar, and we learned a lot about their proliferation rates and mitotic indices. and ultimately their reaction to chemotherapeutic treatments, which is very important when wanting to treat people for cancer. So, thanks for letting us do this research, and I hope you enjoyed our presentation.